Welcome to our channel Mootechie. Today we are exploring the fascinating world of microbial growth and nutrition. We'll be covering key areas like cell division, growth curves and nutrition, topics that are foundational to microbiology. Let's start with the chapter overview, microbial growth and nutrition. This session is all about understanding how microbes replicate and what nutrients they need to power their metabolism. We'll connect the what, the how, and the why behind microbial proliferation. The chapter is organized into three main sections: division, nutrition, and metrics. First, let's get into the fundamentals. Microbial growth is defined as an increase in population number, not the size of individual cells. That's a crucial distinction. It means we measure growth by counting cells, not by how big they get. The primary mode of reproduction is binary fission, where one cell divides into two identical daughter cells. To quantify growth, we use metrics like colony-forming units or optical density measurements. And growth doesn't happen all at once. It occurs in distinct phases: lag, log, stationary, and death, which we'll explore more later. Let's now look closer at cell division and generation time. Binary fission involves several steps. It starts with DNA replication, followed by cell elongation, septum formation, and finally cytokinesis. A key concept here is the generation time G, which is the time required for a population to double. This varies widely by species. For example, E. coli doubles in about 20 minutes, while M. tuberculosis can take over 12 hours. Bacterial populations grow exponentially, which we describe with the equation Nn is equal to N0 times 2 to the power n, where n is the number of generations. It's important to remember that the rate of growth is heavily influenced by factors like nutrient availability, temperature, pH, and oxygen levels. Now, let's talk about the concept of nutrition in microbes. Microbes require macronutrients like carbon, nitrogen, phosphorus, and sulfur for building their structures. They also need micronutrients such as zinc and manganese, which act as cofactors for enzymes. We classify microbes based on their energy source. Phototrophs use light, while chemotrophs use chemical compounds. They're also classified by carbon source. Autotrophs fix carbon dioxide, and heterotrophs rely on organic matter. Nutrients are taken up through mechanisms like active transport, facilitated diffusion, or group translocation. And to grow them in the lab, we use different media types like defined or synthetic media and complex or rich media. Let's examine the interactions between growth and nutrition. Nutrient availability directly dictates microbial destiny. It sets the growth rate and the total biomass a culture can produce. A principle called Liebig's law of the minimum tells us that growth is limited not by the total resources available, but by the scarcest resource, known as the limiting nutrient. We model this relationship using monod kinetics, which describes how the growth rate increases with substrate concentration until it reaches a maximum. When nutrients run low, microbes enter survival modes, like the stringent response, sporulation, or forming biofilms. Balanced growth also depends on optimal ratios of carbon, nitrogen, and phosphorus. Looking at the growth response curve here, you can see that at low nutrient concentrations, the growth rate is very sensitive to changes, but at high concentrations, the rate levels off, following zero-order kinetics. The point labeled Ks is the substrate concentration at which the growth rate is half of its maximum. Why is studying microbial growth so important? Let's look at the real-world impact. In medicine, it's essential for infection control, antibiotic testing, and understanding how pathogens cause disease. For food safety, understanding growth helps prevent spoilage and guides preservation methods like pasteurization and refrigeration. In industry, optimizing microbial growth is key for fermentation processes that produce alcohol, enzymes, and pharmaceuticals. Environmentally, microbes drive nutrient cycling and are used in bioremediation to clean up pollutants. And in research and biotechnology, precise control of growth is foundational for genetic engineering and synthetic biology. From gut health to global ecosystems, the principles of microbial growth have a profound global impact. Here are the key takeaways from today's session. Growth means an increase in population number. It's defined by binary fission and generation time. Nutrients like carbon, nitrogen, and phosphorus dictate the growth rate and yield, and this knowledge is critical for health, food safety, and industry. Don't miss our next deep dive into microbial genetics. Subscribe to Mootechie. If you have any questions, feel free to comment below. I hope this session helped clarify how microbes grow and what fuels their incredible diversity and impact. Remember, these tiny organisms shape our world in enormous ways. Keep questioning and exploring. We'll see you in the next lesson.